and newt at the University of Bern. So Morgane, the floor is yours.
So thank you, everyone. For today, I will not speak about salamanders and newts, but about a project I did last year about birds at the Learning Planet Institute of Paris. So when a bird is singing, it frequently moves its head and neck, and you will understand why it is interesting, because first, if we compared all the species of bird with human, they have impressive frequency capabilities. And this is made possible by an organ named syrinx, just here. So the initial sound is produced by the vibration of membranes and when the air passes. And then it travels through the trachea, a tube formed by a succession of cartilaginous rings. After the larynx, here where you can find the glottis, which is the opening that leads to the trachea, and after the oral cavity. And all this structure represents a significant volume, and their potential movements and deformations have an effect, have a filtration effect on the sound initially produced. We already know that beak and buccal cavity have a significant effect, but the role of the trachea on the sound modulation remains poorly studied. That's why in this study, we want to understand and quantify the impact of the head and neck movement on the trachea shape. But with all these feathers, difficult to see the trachea. So for this, we used X-rays to reconstruct moving morphology. So we made X-ray videos of dead birds moving their head with a stick. We tried to produce movement in as many directions as possible. And we get this type of video on two views to make a 3D reconstruction. We can see, so here the trachea, but it's not visible on whole bird, and we want to be able to follow its movement. So for this, we need markers. So we have implanted a very small metal ball as markers on the head, on trunk bones, and also on the vocal system of the bird. We did this on 16 birds from 16 species belong to 9 different orders to see the deformation, if the deformation of the trachea is the same in all these species. This allows us to obtain this image, as you can see here, of a Picus viridis. I will use this specimen to illustrate the rest of the method. So you can see all the metal balls or the markers implanted in the bird. This allows us to track the marker on 2 cameras that are calibrated in space, and therefore to obtain the 3D coordinates. We need to map the 3D structure to the video. So in parallel, we scanned the bird in order to obtain 3D model after segmentation, and then using a 3D animation software, so Maya, we attach the 3D model to the markers coordinates tracked on the videos. And we have this. So you have the reconstruction of the movement for the trunk, the head, and all the marker here in red for the spine and in blue for the trachea. But even if it's composed of cartilaginous ring, the trachea is not a rigid structure. So I had to find a solution to model its deformation during the movement. And we also noticed that markers were not implanted at equal distance. So, but with the cool thing with Maya software is that we can add whatever we want. So I generated first a curve here from the syrinx to the glottis, passing through each trachea marker in blue. And attached to them, meaning that the curve follow the 3D trajectory of each marker. And then I created 10 equidistant locators along the curve. I used here the word locator to designate a marker added virtually during the 3D animation. This locator will allow us to better study the deformation all along the trachea and to make comparison with the other birds. And for a realistic representation of the trachea, I generated a tube with the diameter of the cartilaginous rings. And here you have the motion reconstruction for the trachea, for Picus viridis. I also had a curve to see the movement of the spin in red. I have applied these steps to a neutral position of the bird, which can be found on the other birds studied here. And I define a coordinate system based on anatomical structures to follow the movement in an equivalent way in all birds. We could observe different things in our results. So first, if we look at the length of the trachea during the motion here of the glottis, we can see a variation of the length. It is higher when the bird's neck is stretched, and lower when it is retracted against the trunk. And we saw the same trend in all birds. Secondly, if we look at the length of the different segments of the trachea in comparison to their length in the neutral position, so all in percentage, we can see that the deformation of the trachea is not necessarily uniform all along the trachea. For some birds, so for example, Picus viridis, the elongation or narrowing is higher at the first curvature of the spin, so here. For other, we can see higher elongation or narrowing at the first curvature of the spin, but also near the syrinx, here or here. And we have two birds with a uniform deformation. Here you have the example of Columba livia. Then we noticed an asymmetrical deformation between the movement to the right and the left of the bird. This for the total length of the trachea, but also for the length of the segment. So this is the case for birds with a non-uniform deformation, of course. Some birds have a higher trachea length and higher segment elongation when the head is on the left side, so for Picus viridis here, or on the right side, here it is the case for Streptopelia decaocto. And this is due to the lateralization of the trachea, because unlike mammals, birds have a trachea located on the right side of the spin for the majority of the species, and for some others, it is located on the left side. And for the left lateralization, this is the case of columbiforms, such as Streptopelia decaocto that we have here in our study. So for an equivalent position on the right and left side of the trunk, the trachea is more stretched on one side, and it will be more elongated where it can be in contact with the spin, for example, here, or here for Picus viridis. So to conclude, we were able to highlight on 16 bird species from 9 different orders the impact of the head and neck motion on the trachea. So an elongation linked to the neck extension, deformation that is not always uniform all along the trachea and which is asymmetric. But how does it impact the sound produced? In parallel, people from the team have shown through simulation on 3D models that 20% shorter trachea results in higher pitch on the frequency. There is a change in the resonating frequency because of the difference of the length of the trachea. So for many of our birds, the trachea elongation is higher than 20%. which would result in a shift higher than this one. And for the moment, I have only talked about extant birds, but because the trachea is composed of cartilaginous ring, it can be preserved during the fossilization process, as here in this fossil birds from the late Miocene of China. And we have also researchers that have been able to reconstruct the hypothetical range of motion of the neck for extinct terror birds from the upper Miocene, lower Pliocene of Argentina. So to better understanding the trachea contribution to sound modulation in extant bird is the first step to infer the potential sound produced in fossil of birds. So thank you for your attention.